Welcome to this biochemistry lecture on amino acids. There are 20 common amino acids that are found in proteins. They are commonly grouped according to property, and this chart kind of has a representation of those groupings generally. It also has the 1-letter code and 3-letter code that are often used to abbreviate the amino acids when we look at polypeptides. This would be a very valuable chart, not only to try to get familiar with, maybe even to memorize, but it's also a handy chart just to keep around in case you have questions. Now that chart is not perfect, it's not a panacea. As you can see from this chart, properties of amino acids are they overlap. And it's not as clear-cut as it sometimes appears. So things like proline and methionine, while commonly labeled as aliphatic and nonpolar, sometimes other people will group them in other categories. And that just kind of depends on what field of study you're in and some of the things that you relate to as a biochemist. So it's just really important to know that this is not always clear-cut. So all amino acids have a very similar structure, with the exception of proline, which we'll talk about here in a little bit. There's an amine group, there's a carboxyl group, there's a hydrogen, and there's an R group, which is the variable part of the amino acids. And that they're all attached to the alpha carbon. Proline, again, is the exception. The amino group forms a ring structure with the R group, and it's actually an amino acid with an I versus an amino acid with an A. So it's really important to note that. The other thing that's important to note here is that this molecule is not flat. The amine group and the hydrogen come out into the screen. The carboxyl and the R group go into the screen, and that three-dimensional structure can have important, and does have important, structure function properties. When we talk about amino acids, we commonly name and number the carbon in particular in order to clarify the situation. So when we talk about amino acids, the alpha carbon is the chiral center of the molecule. And from there we go down to beta, gamma, delta, and epsilon. Lysine is the longest one. Commonly there's not this many carbons in there, but and the carboxyl carbon does not have a letter associated with it, a Greek letter associated with it. But the other ones do. And since they're all a straight chain hydrocarbon, there's no branching in this. It's pretty straightforward when you look at it, which one's the alpha, beta, gamma, and delta. The beta is the first R group carbon. The gamma is the second R group carbon, and so on and so forth. They're also numbered. So the first carbon is, if I'm talking about carbon number 1, it's the carboxyl group carbon. The alpha carbon is actually the second carbon, and then we get into the R groups where we can go 3 up to 6. So let's get into some of the groupings. So the largest group is the nonpolar aliphatic amino acids. It has a couple properties that are pretty important. First off, most of the nonpolar nature of the molecule is set because most of them are straight chain hydrocarbons. There are some branches in there with leucine and valine, but generally speaking, well, there's on several of them there's branch, but generally speaking, we just have hydrocarbons, and that gives it a very hydrophobic property. And so the only two types of interactions you're going to see generally with these the nonpolar aliphatic amino acids are hydrophobic interactions, and that thus they're commonly found on the inside or the core of a protein, and van der Waals forces. And so those are the two kind of things that you'll see. As notes, methionine, which does have a sulfur in it, and proline sometimes are included in this group and sometimes not. So you'll see them sometimes in the polar group, and sometimes you'll see them in the nonpolar group, nonpolar aliphatic. But for this representation, I have selected and put them in the methionine in the nonpolar aliphatic, and that's pretty common. Aromatic amino acids are ring-structured, benzene rings, and so these are very large. Sometimes they include histidine in this group. They have several properties that are very important. First off is generally they're hydrophobic. The tyrosine with the alcohol group does make, and so they're generally hydrophobic. That alcohol group does make it sometimes so that there can be polar interactions, and sometimes the amine on the tryptophan can. But they also absorb UV light around 270 to 280 nanometers, and so we can use this property in various assays to do protein quantification and other related tasks when we want to talk about UV. These are all essential amino acids, meaning we do have to consume them, and they do have different types of bonds. So, and that kind of is dependent on the functional groups. All of them will do van der Waals, especially induced. You will see hydrogen bonding with these occasionally, things with the amine group on the tryptophan, on the hydroxyl group on the tyrosine. You will also see pi stacking with this group, which isn't overly common, but is based on the benzene ring or the phenol ring in this, you will see those interactions. These are commonly found because of their size and charge. They are found commonly on the inside of a molecule, but you'll also see tyrosine and tryptophan in other locations. Polar uncharged amino acids, there are 7 of them. These are those with side chains that they are very polar, so they do prefer to be around water in an aqueous solution, and so you'll find these on the surface of a protein. They will undergo hydrogen and van der Waals forces, and with things like glutamine and asparagine, where you have an oxygen and a nitrogen, you can get localized charges that will allow for multiple bonding to occur. So multiple bonds on the same molecule. In fact, you'll have multiple bonds that are different polar regions on the same molecule. So these interactions are very common and can form hydrogen bonds very, very readily. Next, we have the positively charged amino acids, lysine, arginine, histidine. They actually have the full charge, and so you'll see them very commonly in electrostatic interactions. They are found at binding sites, as are the polar amino acids, but they're also the positively charged amino acids are very large, and they're very susceptible to the buffer. So if you look at histidine, there's no charge on it because that's at physiological pH, but if you change the pH at all, you will get different states of the molecule, as probably noted in different lectures that you had up to this point or in future lectures and notes, and so those are very pH dependent. Negatively charged includes aspartate and glutamate. This has a carboxyl group, which is at physiological pH negative, has a negative charge. Again, these also form electrostatic bonds. These charges are, again, very dependent on the buffer, and they're commonly found at binding sites between protein-protein interactions and other ligands. I would encourage you to spend a little bit of time on this chart, understand this chart, not only know the amino acids that are within these charts, but also the properties and the one-letter code. This common amino acid chart is very handy. Or if you don't have a need to memorize it, you definitely can print this out and use it as a reference. I look forward to seeing you later lectures.